Today I'm going to share mostly a line of research that I've pursued during my PhD. And yeah, it's pretty much along the lines of what Johannes introduced already. I worked on generative models, specifically applied to structure-based drug discovery. But at the very end, I will also lose a few words about the startup that Johannes alluded to. So this line of research that I worked on is how we can apply generative models to early-stage drug discovery. And I want to start by just setting the stage and motivating the problem, even though I'm sure most of you already are familiar more or less what it entails. But still, I think it's always good to be on the same page about the problem setup. So when I talk about drug discovery, I mainly mean small molecules. So that's a molecule like this one. You probably know it. It's ibuprofen. Most of you have probably already taken it at some point. And the general setup that I'm interested in is how can we design such molecules so that it binds to a protein, which is one of these bigger molecules that you might also have seen a few illustrations of in the past. And this is a highly relevant setup because if you look at all the FDA-approved drugs, pretty much all of them are actually small molecules binding to proteins. So yeah, you see 77% are these kinds of small molecules. So this is to set the stage. So this is what we want to do. Let's say the biology is already validated. We know the protein that is involved in a certain disease mechanism. And now we want to interfere with this disease pathway by making a molecule that binds to it and interrupts its function. And the problem why we think we should spend more work and more research on is this one. This is an empirical law. It's called Irum's law that shows you how much money you need to spend in R&D before a drug can enter the market. And note that the axis is a logarithmic scale, the y-axis. So this is actually an exponential decline in the pharmaceutical R&D efficiency that we're observing. And by now, we're spending more than $1 billion per new FDA-approved drug. And one of the core contributors to this effect is this funnel effect. So what I'm showing here in this diagram is the classical drug discovery process, starting from the early target validation, over hit finding, lead optimization. Then you have the preclinical tests, and then later the clinical studies. And your molecules have to pass all of them. And usually you start with tens of thousands of candidates that you're considering in the beginning. And then you run experiments on them. You try to validate if they fulfill all the properties that you're interested in. And with each of these empirical experiments, you lose a few of these initial candidates just because they fail and do not pass the thresholds that you set for yourself. So usually after running through all this pipeline, you end up with a single molecule that then makes it to the market. And the reason why we need to start from 1,000 or tens of thousands of molecules is that biology is extremely complex and we're not very good at predicting what is actually happening once the molecule enters the body. To illustrate this, I'm just showing all the length and time scales here that are involved in the human biology. So if we're talking about atomic interactions, we need to be aware of quantum mechanics, of course, but we also need to be aware of all the dynamics of the process, which span femtoseconds to minutes. So this is many, many orders of magnitude that we would have to model. At the same time, we also have these incredible length scales from the atomic scale on the nanometer level all the way to meters at the organism scale, because you have not a single protein in your body, of course, but you have millions of proteins per cell, and then you have trillions of cells in your body. So to make really accurate predictions, you would have to be good at all these different scales and also connect them in a smart way. So this is difficult to do from first principles. And that's why how drugs are discovered nowadays is what I would call trial and error based, at least in most cases still. So the very common strategy of finding a new molecule that will be further developed into a drug is usually screening based. So you have high throughput screens or DNA encoded library screens, and the exact details don't really matter so much, but you can think of it as just like trying molecules one by one in a functional assay to see if you get the readout that you need. For example, if you have like a diseased cell, if you have some functional effect in that cell. So this is a little bit greedy in a sense, because basically you're first checking if a molecule does the effect that you want, and only afterwards you're trying to figure out how it does it and rationalize why it has this effect. So you're really relying a lot on serendipity. And then, of course, in order to do this well, you cannot just test 10 molecules, but you need to increase your chances of actually finding something that works well. So you actually need to grow these libraries of things that you test. And by now, if you're a big pharma company with a lot of resources, you can test millions of molecules. Or if you're talking about Dell screens, it's even billions of molecules. But even that is only a drop in the ocean compared to all the possible chemistries that you can possibly consider. Okay, so now coming back to this law, this exponential decline, I basically now motivated why it's a bit difficult to rationalize and have a first principle based approach to drug discovery. And next I want to present a potential way forward. And I guess it's a little surprising at an AI symposium that the way forward is actually a computational one, or at least one potential way forward. And for this, I want to contrast this exponential decline in the R&D pharma efficiency to Moore's law, which is this exponential increase of computational capabilities that was empirically predicted by Gordon Moore a couple of years back. And yeah, incidentally, actually, the name Eroom's law for this exponential decline is derived from Moore's law. It's just Moore's law backwards. And I think this is a very striking trend, because basically it is kind of a common thing to, or very natural thing to think that you can counteract one exponential law with the other exponential law. And in fact, we've already seen some examples before. I just have this quick summary slide again of some of the advances that we've seen in this space. We have now machine learning models that are extremely good at docking small molecules to proteins. We have machine learning force fields, and of course AlphaFold, that I'm sure all of you are aware of, which really revolutionized protein structure prediction to such an extent that only, I think, what was it, 3 years after it was published, it already was awarded the Nobel Prize in chemistry. In my own research, I focus on one specific aspect of this whole drug discovery space, which is the initial design setup. And I kind of started this whole thing from the thesis that molecular design is actually something like a distribution learning task or generative modeling task. And by this, I mean that instead of having like a regressor that gives you one single answer, you actually want to sample from all possible answers. And you want to learn from data what this distribution looks like. To formalize this a little bit or abstract this a little bit, the distribution of all the possible molecules can be called P of X, and we want to draw samples from it, X kind of according to the likelihood of these molecules. And this is nice because it's an unsupervised task, so we don't have to spend tons of hours to label data. And we can also get different potential outputs because there's a lot of uncertainty in this process. A single molecule that is predicted to be good doesn't necessarily have to perform well in assays later on. So we would actually like to have several candidates that are all likely and that can all be tested in the lab. And this is a very natural application of generative modeling. So another, basically, the most common strategy right now to generative modeling and the most robust one, it seems, across data modalities are diffusion models. And this is exactly the approach that we also pursued early on. So if you're not aware, I will just very briefly walk through a diffusion model. So basically, you start from a data point. In our case, these are molecules. Yeah, there we go. So this is our clean data point. So this is what it would look like in our data set. It's a structural data point of a molecule. And then during training, we apply varying amounts of noise to it. And we can get noised versions of it. In the beginning, if we just apply a little bit of noise, it will still almost look like the molecule. If we apply more and more noise, eventually the signal to noise ratio will be 0. And then we train a neural network at every noise level to predict the clean structure. And of course, this is much harder at high noise levels and easier at lower noise levels. But overall, once it's trained, we can actually start from a completely random point cloud, apply our neural network several times in a row to stepwise denoise this molecule into the output structure. And this is a very powerful modeling paradigm. So in one of our later instantiations of these generative models that we call drug flow, we actually did this on the whole multimodal structure of molecules, because molecules are not only a continuous pixel value, but they have, well, they do have continuous coordinates, but they also have discrete values on the atom types and the bond types, which define the molecular graph. And additionally, if you want to model the bound pose of a molecule to a protein, you also have to model the bound conformation of the protein itself. And for this, we model the side chain angles, which live on a hypertorus. So this is, again, another domain that has to be modeled simultaneously. And then we have this one denoising model in the middle that takes in all these noisy representations and tries to predict the clean representations for the output. Additionally, we have like a little extra feature in this drug flow model. which is an uncertainty score per atom. So if you ever used AlphaFold, you might have learned to like the PLDDT score, which kind of tells you how much AlphaFold trusts its own prediction. So we have something very similar here, where we assign an uncertainty score for each of the atoms that it predicts, and that can also help users better understand what prediction to trust in and which ones maybe to reconsider. So I told you that we view this as a distribution learning task, so we should actually also evaluate it as a distribution learning task. And one thing that we did to circumvent this problem of evaluating distributions in really high dimensional spaces is to actually just compare distributions of properties of the molecules that we generate to molecules in our training set. So these then become one-dimensional distributions, and for these we can compute Wasserstein distances that we already heard about today, or KL divergences, or whatever distance measure you like. But since this actually results in just big tables, I didn't want to show them here. Instead, I wanted to show something a bit more visual. So what we did for this plot is to project our molecules down into two-dimensional space. We passed them through a pre-trained property predictor. We took the latent embedding of that property predictor and projected it down to 2 dimensions so that each point in these planes represents now one molecule. And the blue points, or the blue shaded area, which is a bit hard to see maybe on the big screen, represents the training data set, and then the black lines represent the samples from the model. And we can see that drug flow is actually a pretty good distribution learner because it covers all these different modes in the training set distributions, at least better than other methods out there. We did a very similar thing for the side chains that we're simultaneously generating of the proteins. Here I'm just showing 3 examples out of the 20 possible amino acids, and then only the first 2 rotameric angles in the side chains. And again, we see that drug flow manages to capture these modes of the distributions quite well. So by this, we can conclude that it's actually quite well behaved distribution learner. But this is often not enough. So to anchor this back into more practical applications in drug discovery, I just have here another representation of this drug discovery pipeline, more or less from start to finish. And what I've talked about so far is basically just this part, learning the distribution of possible molecules, which can be used in the hit discovery stage. And then these molecules would still have to pass through all the downstream preclinical and clinical tests. And a lot of them would probably still fail because we didn't really optimize them for anything in particular. And this is exactly what we would like to do in practice, because maybe there are some properties that we're interested in. A very simple one would be that these molecules should be soluble. And if we call these properties Y, for example, then we can formalize this again using Bayes' rule as actually we want to sample from this probability distribution of P of X given Y instead of the unconditional distribution. And then with a lot of the work that we are also seeing here today and by others that tries to predict properties like that, maybe even all the way up to success in clinical trials, but at the very least functional effects in cells and stuff like that, the better we get, the better oracles we get. If we have a way to feed them back into this design process at the hit finding stage, we can open up this funnel that I showed in the beginning and make success in downstream phases much more likely, just by already rationalizing the design process itself. And there are several ways to achieve this. So this is what we call the goal-directed design states, or we could also call it conditional generative models. And the first one I've kind of showed you already. That's direct conditioning, where you just train your model directly on data points that already have these property labels. Because this is what we're doing when we're conditioning on the protein structure, for example. Another nice way that we can do, especially with diffusion-based models or flow matching models that have these several sampling steps, is to inject some knowledge about the problem or the property at hand in the sampling process itself. And some of the common methods are classifier guidance, particle-based methods, or beam search methods. Another thing we can do is to fine-tune a pre-trained model. So we can first train an unconditional model and then have a second fine-tuning stage where we inject the conditioning information. And a common strategy is preference optimization. I will actually have another slide on that in a second. But before that, I want to mention also a quite more or less disconnected to this general distribution learning literature. There's also the purely reward-guided optimization, where your learning objective itself encodes the property. And the most relevant here is, of course, reinforcement learning that you all know, I'm sure. But there's also a lot of search-based methods that people explore in the drug discovery space. And this is also a very promising line of research. But what we did with RugFlow is actually the fine-tuning approach. This was together with our colleague Adrian. And we kind of took this from large language models. It's how ChatGPT is actually fine-tuned on user preferences. It's the exact same idea. So we start with our reference model. That's just this unconditionally trained model. And then we sample a number of molecules from it. And then we can define any user preferences to define which ones are the good molecules and which ones are the bad molecules. And this user preference doesn't have to be differentiable. It could literally be a person deciding this is good, this is bad. Even though for our benchmarking purposes, we use computational scores. And then we use these two sets, the positive and the negative set, to fine-tune our model, which is initialized with the reference weights of the unconditional model, but then fine-tuned to increase the likelihood of the winning samples while decreasing the likelihoods of the losing samples, and plus some regularization to not deviate too much from our reference model. And this actually works quite well. So what we did here in this experiment is we optimized some computational scores. RIOS, for example, is just some substructure queries that you can apply to your molecules to check whether they're likely to interfere with some assays downstream. And we want many molecules to pass them. And then we can compute the fraction of passing molecules for our training set and for the reference model. And we see, since it's a good distribution learner, they're actually quite similar. And then as another baseline, we also just took the winning examples and fine-tuned our drug flow model just on the winning examples. And this works also to some extent. Now more molecules pass these filters already. But then once we also include the negative samples explicitly in the training, that's the red bar here, we really boost the performance by a lot. And this applies in the same manner also to other computational oracles. We have the empirical drug likeness score, we have a binding affinity proxy, and a synthetic accessibility proxy. And the general trend is pretty much the same across the board. This other bar here, this white and red bar, that's a single preference aligned model, where we aligned this model to all 4 properties at the same time. And of course, it works a little bit worse than specialist models in this case. But what I find interesting is that this combined optimization with negative examples still works better than individual optimizations on just the positive examples. All right, so now basically what I've covered so far is how can we sample molecules, how can we condition these samples on specific properties that we're interested in. But then there's still one major challenge that I haven't discussed yet, because so far everything is just computational models. But if we want to go ahead and actually test if whatever we predict and whatever we design actually works in practice, we have to synthesize these molecules. And that's actually a highly non-trivial task, because we design molecules de novo. It's not clear to a chemist how to make them in most cases. And here, just to show you the landscape, basically, I started in my motivational slides with the high throughput screening and general screening approaches. They kind of live on one side of a spectrum, where the molecules that we get out of these screens are highly available, because by definition, they're already physically available because we screen the physical molecules. But they cover a quite limited chemical space, only what is available for us in our libraries on stock. And on the other hand of the spectrum, we have the de novo generative models that I've discussed now, which cover, in theory, the whole chemical space. In theory, they can operate on the atomic level and generate any bonds. Of course, we're a bit constrained by our training sets, but in theory, they can cover everything. But they're not really available. We don't really know if we have a design how to make it. But luckily, there's something in between, and these are on-demand virtual chemical spaces. These are spaces that are defined by reactions and building blocks that it's often commercial companies, but also sometimes in academia, that you just have in stock. You know you have these building blocks, and you pre-validate how these building blocks react with each other. So you have some really robust reactions. You know you get high yields and high success rates out of these reactions. So you know, in theory, what goes with what. But you never fully enumerate. You never have like a list on your computer with all the molecules that you can store. But the space is more like implicitly defined by the sets of building blocks and reactions. And then the computational task kind of becomes how to efficiently search the space, which is only implicitly defined. And again, there are several approaches that people took and that are actively pursued, actually. The perhaps simplest one is to take a designed molecule and project it onto this synthesizable on-demand space. Basically, you just look for the closest analog, where the crux is to define closeness appropriately. And then you order that molecule instead. You get that molecule synthesized instead of your original design. But there's a clear disadvantage. You're not actually ever testing your actual designs, just something that looks similar to them. So another strategy is to do retrosynthesis planning. So you start from the molecule that you designed, and you have some neural networks or even other approaches that try to devise a synthesis route that could work. So you take, in the simplest case, you just have a molecule, and you try to predict the precursor molecules that you need, the reagents and the reactants that you need, that you then put in your test tube and get an output, get the reaction running, and you get your product molecule out. This, of course, is also still limited because there's basically no guarantee that if you apply these algorithms to your molecules, that you actually end up with starting materials that you have available, or that the reactions are feasible, or even if they're feasible, that they can be run in just a couple of steps and you don't have to spend like 1 year of more R&D on it. So what we try to do instead is a more bottom-up approach, where we try to bake in the synthesis constraints already in the design process. And for this, I have to briefly introduce the successor, the next version after drug flow that we trained, because we applied a little twist at training time. We call this the large drug discovery model, or LDDM for short. And under the hood is still pretty much drug flow. So this lower panel here is still the exact same as what I showed before with drug flow. But now we have this additional training trick where before we train the model to generate a molecule, we actually first fragment the molecule. We define little substructures. It's a bit hard to see because the background colors are not very clear on the big screen. But basically, these are different fragments here. And then at training time, we apply random mask, a bit similar to how large language models are trained by masking out tokens. But we instead mask out these fragments and ask the model to generate what is missing in every step. So now the nice thing about this is that now the model can actually condition on molecular fragments in addition to just a protein pocket. And so this is something we can then embed in a sampling algorithm where we iteratively update our molecules. So in the first iteration, it would still look the exact same way as drug flow. This is supposed to show an empty protein pocket, and it designs a number of molecules just from scratch in an unconditional way. But then instead of just taking these molecules and taking them as our final designs, we look for substructure matches with our available building blocks. And we take only the parts that we find in our building blocks and remove all the rest, and then repeat this process. We condition LDDM in the next iteration on these fragments to generate a new generation of molecules that all contain these substructures. And then in the second generation, we repeat the process. But now we don't only look for molecules that resemble building blocks, but now we actually check, can we go from the first building block with a reaction, our reaction database, to this substructure in the second generation. And if we find it, we're happy, we remove everything else, and we repeat this process until whatever success criterion we defined is met. And this is cool because now we don't only have molecules out with their proposed binding pose, but we also get a proposed synthesis route with every molecular design. And this actually allowed us to go to the lab and test some of these designs that we made. And I just want to quickly show 2 case studies. The first one is on a protein called PGK1. And for this one, we designed thousands of molecules. We applied some additional filters and ranked our molecules, and we ordered the 6 ones that look best according to our computational metrics, which are shown here on the left-hand side of the slide, with the best candidate shown in the predicted binding pose here on the right-hand side of the slide. And then we went to the lab. We took these molecules. We ordered them from one of these on-demand spaces, and we tested them in biophysical assays if they bind to the target. And for 4 of these 6 molecules, we did actually observe some binding. Those are the ones shaded in blue. We also computed the binding affinity based on a multi-cycle kinetics experiment. And for the best molecule, which is the one shown on the right here, we got 15 micromolar binding out of this. And then we took them to an NMR spectroscopy experiment to also verify that we're actually binding in the right binding site. Because this is a 2-domain protein. In theory, if you get unlucky, your designs could also bind down here, for example, in this lower domain. But we could see by the spectral shifts in the NMR that we're at least targeting the right domain. We still don't have a crystal structure to fully confirm the binding mode, unfortunately. The second case study is on another protein that's called KRAS. In this case, we managed to synthesize 5 molecules, and 3 out of them showed some binding signal. And again, we could also measure the binding affinity in the lab. And in this case, it ended up being around 86 micromolar. So for the ones of you that are actually familiar with these kinds of binding affinities and what you would expect for a drug candidate, you'll realize this is not something you would take to any clinical trials. They bind way too weakly. You would expect much stronger binding for a real drug candidate. But it does fall into the category of hit molecules that you would also get out of a high throughput screen, for example. But nevertheless, I would argue that this is still one of the main remaining challenges, because the whole premise of the computational drug discovery approach is that we can actually do a lot of the iterations in the computer, and we don't have to go to the lab and run many rounds of optimization anymore. So we definitely need to improve these binding affinities that come straight from the computer. And one of the reasons why we're not better, in my opinion, I think, or I would venture to guess, is that these molecules are very multimodal. So the generative setup is actually not so easy to do. I showed you one of our approaches with the drug flow model that does this multimodal generation, but there's actually lots of knobs to tune that we didn't fully hyperparameter tune. So for example, you have these denoising schedules in diffusion models. And now for every modality, we have a separate denoising schedule. And the interplay between these denoising schedules is not trivial. And it's extremely hard to tune because it's actually a continuous hyperparameter. Another thing that's, I think, also very obvious to anyone working in the field is that we are in a very data scarce domain. So if you're just thinking about crystal structures from the PDB, there's around 50,000 data points. And if you're actually focusing on the ones that are relevant that look like drugs and they duplicate also a little bit, you actually end up more in the realm of 10,000 data points roughly. And in addition to that, they're quite noisy because there's modeling errors. Sometimes the resolution of these crystal structures is not very high and people just put atoms where they think they should be, even though there's actually no experimental proof that the atoms are actually there. So there's a lot of noise in this. And if you think or compare the small molecule task to a protein design task, this noise is actually more important. Because if you're designing a protein binder, for example, you have large interfaces. So there's a lot of atoms involved in the interaction. So if one or two of them are not accurately modeled, maybe you can get away with it because there's so much more signal that can outcompete the wrong model. But in small molecule binding sites, you have much less atoms involved in the interaction. So basically any wrongly placed atom can already be completely detrimental to your binding. And then the last challenge that we have to consider in this space, and the previous speakers have also already alluded to this, is that we kind of need to be smart here because the data is not huge. We cannot just apply the exact same LLM scaling strategies that work in other domains, or just blindly apply transformer is going to give you the best results. But we actually need to be a bit smart. We might have to think about inductive biases that make our prediction easier. And to visualize this, I actually wanted to contrast this plot that I took from a Hugging Face blog post of how LLMs were scaled over the last years to the largest model, the large drug discovery model that I showed in this presentation, which would live right down here at the bottom line of this plot with around 10 million parameters. And we have never observed any positive effects by scaling this more. All right, with this, I'm already at my conclusion slide. My main takeaways from my research and my PhD were that diffusion models are actually quite robust and a great generative modeling framework for pretty much any data modality that I'm aware of. They seem to be state of the art in video generation, image generation, and apparently also for molecules right now. They're also nice because they're flexible. You can very easily turn an unconditional model into a conditional model and condition on different properties that you're interested in. And if you then also bake in the synthesis constraints into the process, you can actually go to the lab and close this feedback loop, which I think if more and more people do this in the future, we will also make a lot more progress on the computational side because we actually get valuable real-world feedback from this. And then, yeah, this is what I alluded to on the last slide. I really think it's an interesting domain to work in because we need to be a bit smart. We cannot just blindly apply what works in other domains, but really domain expertise is crucial to make progress here. Yes, and with this, I want to thank you for your attention. I want to thank my advisors, Bruno and Michael, but also my collaborators that worked on all these projects that are presented here today with me, and the whole laboratory of protein design and immunoengineering. Yes, and before I take questions, I also wanted to very briefly mention, I mean, Johannes already mentioned it, now that I moved on from my PhD, I'm actually working on a startup. We call ourselves Biolabs, and we're kind of continuing this exact line of research that I presented here today. So if anyone is interested in collaborations or whatever, is just interested in what we're doing, please reach out.
Fantastic. Thank you very much for this talk. Who would like to ask the first question?
Thank you for your presentation. I was wondering, have you tried sequential Monte Carlo-based method for sampling from this conditional posterior distribution? And if yes, how well does it work?
We have tried. It's a bit finicky. So what we did, and correct me if you had something else in mind, but we took these diffusion models and we basically, our proposal in the sequential Monte Carlo was generated by noising a molecule a little bit and then denoising back. So basically you get a derived output from the original starting point this way. And this works well. It just also has lots of knobs to tune because this level that you noise and that you get back, then you have the acceptance criterion which you need to tune. And we never found it to work better than just sampling from a Gaussian all the way to clean data, unfortunately.
Thank you very much.
Other questions? Well, there's one there.
Thank you for your presentation. I have a question about the fine tuning via the preference optimization. I know exactly the process of your like data augmentation tasks, but I'm just curious about how you train and fine tuning your preference model. I mean, maybe in two different aspects. First, how you prepare for your data? Is it like a human annotated data sites? And the second is what is the backbone of your preference data model? Yeah, thanks.
Yes. So in theory, as I mentioned, you could really go ahead and label data, and this is also something we're considering to do with the startup actually, because once you scale up things and have chemists using your tools, you can collect that feedback from users to kind of extract some expert knowledge that is otherwise really hard to codify, right? But if you can just have a chemist tell you, yes, this is great, this is not great, you can learn a lot from that. In the context of this research project, this was not feasible for us because you would have to collect hundreds of thousands of these responses for this to make any sense in practice. So what we did instead is we just used computational oracles. So basically on these filters, for example, Rios filter is really just a binary thing. It either passes it or it doesn't pass it. So we could really use that as a preference. A good molecule is one that passes, a bad molecule is one that doesn't pass. So this is a very natural setup for this learning scheme actually. And this is probably also why it works the best on that particular metric. For other metrics like the QED, it's a drug likeness score. It's just a very fuzzily defined way of telling you how drug-like a molecule looks. That's the continuous score between 0 and 1. And we just randomly, more or less arbitrarily defined a threshold at 0.5. And we said, okay, everything above that is good, below is bad, but it's actually inherently a continuous value. So maybe this is also why it doesn't work as clearly well as for the Rios filter. And then to the second question, our model is a GNN. It's a geometric GNN. It's E3 equivariant, which means if we change our input, we rotate it or we translate it, we will get the exact same output, just also rotated or translated, which we found to help in practice because we have so little data points, and then we don't rely heavily on data augmentation of that kind, but instead we can really just focus on like an efficient model. Maybe on the topic of GNN, it's maybe not the most traditional GNN in the sense that we connect all the nodes with all the other nodes. So maybe in some sense it's even closer to a transformer, but we still call it GNN because it is message passing based, what we do.